Dear students, today I will discuss on enzyme-linked immunosorbent assay or ELISA. It is an immunoassay which is routinely done in healthcare, in scientific research, also in environmental and agricultural applications. And this immunoassay involves the use of an immunosorbent. That means an antigen or an immunoglobulin is adsorbed on a solid surface. Then we add an analyte. Analyte means the sample which we want to test, and then we add an enzyme linked antibody and detect the presence of the analyte. So the components of ELISA are antibody or immunoglobulin, analyte, enzyme and its specific substrate. Antibodies or immunoglobulins are gamma globulins, and they are generated in jawed vertebrates in response to a foreign molecule or immunogen. And each antibody molecule is composed of 4 polypeptides. 2 of them are long and 2 are small. The long chains are called heavy chains and the small chains are called light chains. And the sequence of this region varies among different immunoglobulins. So this region is called the variable region. And actually the variable region binds to an antigen. So this is the antigen binding site, and each antibody molecule has 2 antigen binding sites. And the sequence of this region is to some extent constant. And if we characterize all the immunoglobulins present in our serum, then we will see there are 5 different types of sequences present in the Fc region. So this region is called constant region or Fc region. Now the antibody which binds to an antigen is called a primary antibody in immunoassays, and an antibody which binds to the Fc region of a primary antibody is called a secondary antibody. And an enzyme or fluorescent dye is tagged with the Fc region of a secondary antibody. Normally, horseradish peroxidase is the enzyme of choice, and its specific substrates are trimethylbenzidine and hydrogen peroxide. When these 2 substrates are added to a reaction mixture having horseradish peroxidase tagged antibody, the HRP enzyme will convert H2O2 into H2O, that is water, and nascent oxygen. The nascent oxygen will oxidize trimethylbenzidine to trimethylbenzidine oxide, and it will give a coloration. Similarly, if we use alkaline phosphatase as the enzyme, we have to use its specific substrate paranitrophenyl phosphate. Okay? Alkaline phosphatase will cleave the phosphoryl group and it will generate a color. Now, ELISA is becoming very popular because of its sensitivity. It can detect minute amount of an analyte present in a sample quantitatively, and ELISA assay kits are available in market. Now, if we look into the sensitivity of immunoassay, then we can see that ELISA can detect as low as 0.01 nanogram per ml sample or analyte present in a sample. That means it has comparable sensitivity with radioimmunoassay. Radioimmunoassay was the only immunoassay used for conducting different immunoassays before the invention of ELISA. But since radioimmunoassay involves the use of radioactive material, it is not recommended very often. So ELISA is becoming very popular since it is using enzymes instead of radioactivity. ELISA was discovered by Peter Perlman and his graduate student Eva Ingvall at Stockholm University in Sweden, and by Anton Scootz and B.V. Wieman in Netherlands at the same time in 1971. Now, ELISA have been developed further, and now various types of techniques are available like direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA, and very recently new technique of ELISA have been developed. It is called multiple and portable ELISA. This is the simplest of all ELISA techniques. And how we can perform direct ELISA? First we have to add the antigen sample, both standard and test sample, in microtiter wells, and we have to give some time for adsorption of the sample to the microtiter well. Now microtiter wells are made of polystyrene or polycarbonate material, and the antigen which we want to analyze is normally a protein which will bind to the polystyrene material or polycarbonate material through non-covalent interactions, mainly through hydrophobic interactions. Clear? Now we have to wash the wells properly. After washing, we have to block all the free spaces present in a microtiter well, because we want only specific interaction between the antigen present in the sample with antibody. Normally bovine serum albumin or ovalbumin is used to block the non-specific sites. Then again, after washing, we have to add antibody tagged with an enzyme directly. This is why this technique is called direct ELISA. Here we are directly detecting the presence of the antigen using an antibody tagged with an enzyme. Again, after proper incubation time, we have to wash the wells. Then we will add the specific substrate. After some time, say after 10 minutes, we have to stop the reaction with either alkali or acid. This stopping of the reaction is very important, because since enzymes act as an amplifier, if only few enzymes are present in a well, they can convert all the substrates into products. Clear? Then we have to see the coloration. Now can you tell me why we are washing the wells after each addition?
Yes, ma'am. I suppose if excess of antigen will be present in the sample, then it may give a false result. So hence washing is now good.
Yes, we want specific interaction between the antigen and antibody. That is why we want to make sure that only the bound antigen and bound antibody remains in the well. That is why we have to wash the wells after each addition. Okay. Now color intensity is measured in an ELISA reader. The extent of intensity depends on the amount of antigen present in a well. These are the wells looking white. That means no antigen is present in this well. Whereas these wells contain less antigen. And these wells, intense dark color means amount of antigen is more in these wells. Then we have to measure the intensity of each well through an ELISA reader, and we will plot the standard antigen values with their corresponding absorbance values. Then we plot a standard curve. Each time we do an ELISA, we have to draw a standard curve, and from the standard curve we can find out the concentration of the antigen present in our sample. Direct ELISA has many advantages. Since only one antibody is used here, the technique is very simple and fast, and since only one antibody is used here, cross-reactivity between different antibodies can be eliminated in this technique.
Ma'am, can you please explain the phenomenon of cross-reactivity?
Yes, cross-reactivity is actually when an immunoglobulin raised against one antigen reacts with a different antigen. Okay, and it happens between antibodies raised in different species. Now disadvantages are also there in direct ELISA, like since the antigen is directly tagged with the primary antibody, each time we have to tag the enzyme with the primary antibody. So it is time consuming, at the same time expensive. And immunoreactivity of the primary antibody may be hampered by the labeling. The signal is less amplified in direct ELISA. Indirect ELISA is a two-step process. Two antibodies are used here. One is primary antibody and the second one is a labeled secondary antibody. Now how we can perform indirect ELISA? First we have to add the antigen in a microtiter plate. Then we have to wash the wells properly to eliminate all the excess antigen. In the next step we will add the sample. if the sample is carrying specific antibodies against this antigen, they will bind to the antigen. We have to then again wash the wells so that excess antibodies or unbound antibodies are washed off from the wells. Next, we will add an enzyme tagged secondary antibody specific for the primary antibody used here. And after washing and incubation, we will add the substrate, and just seeing at the color change and intensity of the color, we can find out whether the specific primary antibody is present in the sample or not. And if it is present, then from the color intensity we can find out its amount. Now, since two antibodies are used here and the presence of the primary antibody is detected by using a second antibody or secondary antibody, this process is called an indirect ELISA. Now, there are many advantages of indirect ELISA, like many primary antibodies generated in one species can be detected by using a single enzyme labeled or fluorescent dye tagged secondary antibody, because secondary antibodies are developed against the Fc region of a primary antibody. And a wide variety of enzyme linked secondary antibodies are available commercially, and this reaction is highly sensitive because we are using more than one antibody here. And immunoreactivity of the primary antibody is not hampered because we are using the enzyme tagged secondary antibody. What are the disadvantages of indirect ELISA? Since we are using two antibodies, the chances of cross reactivity increase in this technique, and since the technique uses more than one antibody, obviously it will take more time. So, extra incubation step is required here. This technique involves two antibodies raised against two different epitopes of the same antigen, and antigen is trapped between the two antibodies. That is why it is called sandwich ELISA. How we can perform sandwich ELISA? First, we have to coat the microtiter wells with one antibody. This antibody is called capture antibody and it is not labeled. Next, after washing the wells properly, we will add the antigen. If the specific antigens are present in the sample, they will bind to the antibody. Then again we have to wash the wells properly to remove all the unbound antigens. Next, we will add the enzyme tagged second antibody. Remember, this is not a secondary antibody. Secondary antibodies are generated against the Fc region of the primary antibody. Here the second antibody is generated against a second epitope of the same antigen. Now, if the sample is carrying the antigen, then the sandwich will be formed, and just upon on the addition of the substrate will change its color, and we can see whether the antigen is present in the sample or not. And quantitatively we can detect the presence of the antigen by measuring the intensity in an ELISA reader. And this is an example of direct sandwich ELISA, since the antibody which is directly bind to the antigen is tagged with an enzyme. Here we use total of 3 antibodies, one capture antibody, antigen, a second antibody which is raised against a different epitope of the same antigen, and presence of this antibody is detected by using a secondary antibody tagged with an enzyme. So, this is an example of sandwich ELISA, but indirect sandwich ELISA. Now, this technique has several advantages, like it is highly specific since at least two antibodies are used in this technique, and it is suitable for complex samples because the antigen does not require purification prior to measurement. We can directly add the sample, and flexible and sensitive since both direct and indirect detection methods can be used here. the antibodies specific for an antigen are incubated with the sample first. So, initially we incubate the immunoglobulins with the sample. Now, if the specific antigen is present in the sample, they will bind to the antibody. Now, this mixture is added to a well coated with antigen. Now, if the antigen concentration is high in the sample, then there will be very little free antibody available to bind to the antigen coated in a plate. Then after washing, we will add a secondary antibody tagged with an enzyme. Then again after incubation and washing, we will add the substrate. Now remember one thing, in competitive ELISA, less coloration means more antigen present in the sample. Less coloration means less enzyme tagged secondary antibody present in the microtiter well, and that is only possible when there will be less free antibodies available, and that is possible when the sample is carrying high amount of antigen. Clear? That is why when incubated with the antibody solution, all the antigen binding sites are filled up with antigen. And this method is also highly sensitive since two antibodies are used to detect the presence of the analyte, and it is also suitable for complex samples. This technique does not require purification of the sample. Now I will tell you a new technique, new ELISA technique, which is called multiple and portable ELISA. This technique uses a solid immunosorbent polystyrene rod with 8 to 10 protruding ogives or pin-like structure, and each of them can be dipped in a tube containing sample. Then after incubation and washing, this rod is immersed in conjugates carrying antibodies. Again after incubation and washing, the rod is immersed in chromogenic substrate, and immediately color change tells us the presence of the antigen in that sample.
Ma'am, what is chromogenic substrate?
Chromogenic substrate is a molecule which changes its color after oxidation, so that we can very easily see the change in color. Now, multiple and portable ELISA has multiple advantages, like each rod can be sensitized to a different reagent, and thus at the same time we can detect the presence of multiple analytes. And since washing steps are very simple here, we can do the assay very quickly and on the site where we want to perform it. Advantages of this ELISA is that each rod separately can be sensitized for different antibodies. So at a time we can perform many different experiments, and washing then addition of reagents is very simple here, so it can be done very quickly and on spot. ELISA has multiple applications, like it is used to measure the presence and amount of antibody in our serum, like in case of allergy or in case of autoimmunity. Also it is used to detect the presence of viruses in blood, like HIV or hepatitis viruses. Also we can detect the change in hormonal levels, mainly human chorionic gonadotropin hormone, which is a marker of pregnancy, can be detected, and many ELISA kits are available in market to detect the pregnancy readily using ELISA technique. Also we can detect the thyroid hormones very easily through ELISA. Also we can detect the presence of toxins in contaminated food or presence of cocaine in blood. So ELISA has multiple applications. So today we learned the immunoassay technique, enzyme linked immunosorbent assay. What is the basic principle of this technique? How it works? Okay, what are the different types of ELISA? We know now that there are 5 different types of ELISA, and advantages and disadvantages of each type of ELISA and its applications.
you.